about sticking a gene for making a protein into cells to get cells to make the protein for us. But if we were to actually stick in the gene, the cells would be like, what's all that extra info? I can't make a functional protein from this. So instead, what we stick in is the CDS, the coding sequence, which is part of the cDNA, the complementary DNA, which is a DNA copy of the messenger RNA, which is an edited version of the pre-messenger RNA, which is an RNA version of the gene. The major difference between the CDS, so the coding sequence, and the cDNA, the complementary DNA, is that the complementary DNA includes the UTRs, the untranslated regions, so the 5' UTR at the beginning and the 3' UTR at the end. So hopefully you can forgive us for saying gene, but also hopefully by the end of this video you can understand why it matters that we do have all these terms to make these distinctions. So let's step back and think about what we actually mean by gene. Genes are regions of DNA that have the instructions for making a protein. This includes the regulatory regions as well as the regions that actually have the information for placing amino acids in a specific order to make a specific protein. Genes are transcribed into pre-messenger RNA. So you make a letter-for-letter copy of the DNA. We call this the pre-messenger RNA. And then that gets processed to give you the mature messenger RNA. It can get processed in different ways, as we'll talk about, to give you different versions of the messenger RNA, different like spliced isoforms. We can reverse transcribe that messenger RNA and basically take it from RNA back to DNA, and that is going to be how we make complementary DNA or cDNA. So the cDNA is going to contain everything that was in that mature messenger RNA. And that mature messenger RNA includes the exons, the parts that were actually expressed, make it into that messenger RNA. It does not include the introns, the intragenic regions that kind of break up the exons and allow for things like alternative splicing. Those introns get transcribed, they make it into the pre-messenger RNA, but then they get removed before you get that final messenger RNA. And so that mature messenger RNA includes the exons, but not the introns. When we think about exons, we often focus on the parts that have the instructions for placing amino acids in a specific order. But exons also include the 5' UTR at the beginning and the 3' UTR at the end. UTR stands for untranslated regions. Because these regions aren't translated, they're not kind of made into protein. They don't have the instructions for placing amino acids. Instead, they have things like sites for ribosome binding, sites for regulation through microRNAs and things. And so these are important bookends to the messenger RNAs, and thus they are part of the messenger RNAs, and thus they're considered exons, but they're not actually having the amino acid placing instructions. And these 5' UTRs and the 3' UTRs would be part of the cDNA, but they're not part of the CDS, so the coding sequence. The coding sequence is the part that actually encodes for the amino acids, and so it has the codons, which have the instructions that say, place this amino acid here, then here, then here, then here, to get this final protein. And that coding sequence is the only part that you want to actually give to cells if you want them to make the protein for you. For a few reasons. One is that if you were to stick in the part with still the introns into cells like bacteria, the bacteria don't actually have the splicing machinery in order to carry out the splicing. If you were to stick it into cells that did have that splicing machinery, they still wouldn't know which splice isoform to form, so which version of it, because you can remove different exons to form different versions or splicing isoforms of the messenger RNA and then of the protein. And so you need to stick in the CDS for the protein version, the isoform that you actually want. And you don't want to stick in those UTRs and stuff because the cells, when you stick it into basically a plasmid, to stick it into cells, you typically kind of replace those regulatory regions on either side with regulatory information that's actually in the plasmid itself. So in sort of the piece of DNA that you stick into the cells, so that the cells can optimize the use of their own machinery in order to make protein from that gene that you stick in. But remember that the gene is not actually a gene that you're sticking in. Instead, you're sticking in the CDS, the coding sequence coming from the part of the mature messenger RNA. So part of the messenger RNA that actually had the instructions for making a protein, but we're sticking in the DNA version of that, and so we call that the CDS. And so it's the part of the cDNA, the complementary DNA, that actually had that information that says, place this amino acid here, 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 here. And so now you might be wondering, okay, well, how do I find the CDS? One of the ways you can do this is if you go to UniProt, which is kind of this database which has all the information about proteins, including the sequences of the different splicing isoforms and things. And so and other sorts of isoforms. So if you go to UniProt and you go to the sequence part, you'll see that you see different isoforms listed potentially. The top one is typically like the main canonical one, and everything else is compared to that. It doesn't necessarily mean that it's the most common one, but it often is. You can like click down and see the different isoforms, and some of them are kind of like legit ones, and some of them are computationally predicted, which might be true, and they might not actually be made. If you go down, you'll see something with the cDNA. That is the consensus cDNA database. And there they have the actual DNA sequences that have the instructions for making those isoforms that you see listed in UniProt. Because UniProt is going to show you the amino acids, and then if you want the DNA that encodes for those amino acids, so that is, you want the cDNA, you can go to the cDNA library. Now say you want to actually go and make the protein. Now you need to find a source of that DNA. So you can do things like use a tool like AdGene and see if they have it cloned in a vector already. You can use something like DNASU. So it's another plasmid repository that has a bunch more of these sequences. Or you could synthesize it. So now that you have the sequence, you can send it off to some company to make it from scratch for you. Or you can use a cDNA library where scientists basically take all of the messenger RNAs in a cell, they reverse transcribe them into complementary DNA, cDNA, and they stick those into plasmids so that you can make lots of copies with them, and then they host those. And then what they can do is that if you have a sequence that you know that's part of the gene of interest or the cDNA of interest, you can use that as a probe to kind of like fish out the cDNA that you want, and then you have kind of the whole version of it that you can get that way, and then kind of subclone it, take it out of that vector and stick it somewhere else you want to work with. And so there are different options depending on how common the thing you want to work with is, how long and how complex it is it more expensive to synthesize it, that sort of thing. But bottom line, we put the cDNA into cells to make the protein for us. The cDNA is the coding sequence, which is part of the cDNA that does not include the 5' and the 3' UTRs, but it does include the parts that actually have the amino acid placing instructions. The messenger RNA is an edited version of the pre-messenger RNA, but the messenger RNA includes the 3' and 5' UTR, which are still considered exons because they're taken from the DNA into the pre-messenger RNA. The introns are intragenic regions that are part of the gene, they're part of the DNA, and they make it into the pre-messenger RNA, but then they get cut out. So the introns aren't typically part of your cDNA because they're not part of the mature messenger RNA. But the 5' and the 3' UTR, they still are part of that messenger RNA, the mature version. They don't get spliced out because they're important for telling the cells and things how to make the protein and regulation of it. But those actually aren't important when you stick it into a different context with these cells, like bacteria that don't actually know what to do with that extra information. And so you're kind of placing just the coding sequence that you want with the regulation information from the bacteria on either side of it when you stick it into something like a plasmid. So hopefully now you understand why we technically stick the cDNA, and it's important to know that distinction, but you'll also forgive us for just saying gene, because sometimes this jargon can get incredibly dense.